Welcome to this video on applied bioinformatics. We will explore how complex biological systems are translated into computational data through various molecular techniques. In this video, we look at 4 lenses of molecular resolution: DNA barcoding for single species, metagenomics for entire ecosystems, WGBS for the epigenome, and sequence searches. Defining an ideal DNA barcode requires 3 key criteria: significant species-level variability, conserved flanking sites for universal primers, and a short overall sequence length. The barcode production pipeline consists of 4 main steps: sample collection, sequence amplification using PCR, barcode generation through sequencing, and comparison against the bold library. Metagenomics allows us to look beyond the petri dish. By studying microbial communities directly, we gain insights into diversity, ecological relations, and bioprospecting opportunities. Metagenomic analysis can be targeted using specific markers like 16 srRNA or comprehensive through random shotgun sequencing of total community DNA. Epigenetic markers such as 5-methylcytosine play a vital role in regulating gene expression by modifying DNA regions without changing the underlying genetic sequence. Bisulfite conversion is a key mechanism for identifying these marks. By treating DNA with bisulfite, unmethylated cytosines are converted to uracil, eventually appearing as thymine after PCR. The whole genome bisulfite sequencing workflow moves from the wet lab, where chemical preparation occurs, to the dry lab, where bioinformatics tools analyze the resulting sequence data. The NCBI ecosystem provides essential resources. Tools like PrimerBlast assist in designing PCR primers, while databases like dbSNP store information on genetic variants. To quantify alignment confidence, we use the E-value. It represents the number of matches expected by chance, with values closer to 0 indicating higher reliability. Precision in primer design helps avoid false positives. By spanning exon-intron boundaries, we ensure that only expressed RNA, represented as cDNA, is successfully amplified. Clinically, we can detect specific variants like 1,800,571 rupees. Using PCR-FLP analysis, we can determine if a genetic mutation alters a restriction enzyme cut site. This matrix summarizes our bioinformatics solutions, mapping various biological questions to the appropriate target scopes, laboratory techniques, and computational tools. In summary, DNA barcoding relies on high divergence, conserved flanking sites, and short lengths to effectively identify species, whether animal or plant. Finally, the WGBS bioinformatics pipeline processes converted sequence data through 5 key steps: alignment, MEC calling, depth analysis, methylation quantification, and global methylome trend characterization.